Hi everyone, my name is Yoann Anselmetti and today I will present you the work done by me and my colleague that has been interested in the designing of an algorithm and a method to reconstruct the evolutionary history of endosymbiotic gene transfer in the context of gene tree and species tree reconciliation. First, I will introduce you the biological context in which this research has been done. So it is well known that mitochondria originate from the integration of an endosymbiotic alpha proteobacteria, represented by the red line on the figure, into an archaeal host cell, represented by the purple line. And this endosymbiosis lead to the emergence of eukaryote cells and to the eukaryote cells. Across the history of evolution of eukaryotes, mitochondrial gene content have been shaped by early massive gene loss due to repeat gene function in between mitochondrial genome and nuclear genome, and also across evolutionary history by episode of endosymbiotic gene transfer that correspond to gene transfer between the mitochondria and the nucleus genome, and this result in a significant reduction of the mitochondrial genome that leads to also loss of function in aerobic mitochondria and give rise to different categories of mitochondria from the aerobic to the mitosome, and same in some lineage of eukaryotes, you have the completely lost of mitochondria. So we understand that endosymbiotic gene transfer is a very important evolutionary process to understand the evolutionary of eukaryotes, but until now, there is no method to reconstruct the evolutionary history of this important evolutionary event. So in this study, we were based on the method based on gene tree reconciliation with species tree reconciliation, and more precisely duplication and loss reconciliation. So it takes as input gene trees that represent evolutionary history of gene families, a species tree, and they will consist to reconcile the topology of the gene family gene tree into the topology of a species tree by inferring evolutionary events that are duplication and loss. And so we lead to a reconciled tree in duplication and loss. Here that infer ancestral duplication C3 in the ancestral species C, and there is a lost B of gene B1 in species B. And here is different representation of the reconciled tree. So if we want to take into account the endosymbiotic gene transfer event, we need to extend this DL reconciliation. First, we need to consider a new parameter that is a genome location to say if a gene is located on the mitochondrial genome, that is represented by S0 for species L. 0 means that it's located on the mitochondria, or S1 that represents the nuclear genome. After that, we also need to have two new evolutionary events. There is the EGT cut and the EGT copy. EGT cut represents endosymbiotic gene transfer, where a gene comes from a genomic compartment, here mitochondria, and go in the nucleus compartment. And in the EGT copy, you have a duplication. So the copy in the mitochondria remains, but there is a new copy that is located after in the nucleus. So the DL reconciliation problem take as input a DL reconciliation of a gene tree T. So the DL reconciliation is represented below. We have also a species labeling that represent for each gene to which species it belongs in the species tree, and the genome labeling that represent if the gene is located in the nuclear or in the mitochondria. 0 for mitochondria, 1 for nucleus. And there is also a cost function on duplication loss and EGT that consider so the speciation, duplication, loss, and EGT copy and EGT cut. And after, it leads to a most parsimonious DLE-reconciliation that reconciles the evolutionary history of a gene family with transfer evolutionary endosymbiotic gene transfer. And so at each node of the gene tree, we have the localization of the gene if it's located on the mitochondria or in the nucleus. So the algorithm we developed is divided in 3 steps. First step consists to define the ancestral species labeling. So this step is made with LCA mapping, and we show in the paper it is correct to use LCA mapping for species labeling in DLE-reconciliation. We also use we have a second step that consists to define the possible ancestral type labeling. So this is equivalent to the bottom-up algorithm of a modified version of Fitch algorithm, I will present after. And there is a third step that is correspond to a top-down algorithm that is so the Fitch remaining algorithm that consists to, from possible ancestral genome labeling, define the correct ancestral genome labeling and infer the endosymbiotic gene transfer that will minimize the DLE reconciliation. So in this presentation, I will focus on the bottom-up algorithm and top-down algorithm. So bottom-up algorithm, as I said, it's a Fitch modified. So in the Fitch consists to reconstruct the labeling in a parsimonious method in a tree where we have a label at the leaf and to find the correct labeling that minimize the number of permutation between labeling. Or here we have 2 labeling, just O or 1. So from an ancestral node, we take the right and the left children, and if the assignation of left and right children is empty, in this case, in Fitch, we take the union of the 2 assignations. If there is an intersection between the assignation, we take the intersect. But we modify the Fitch algorithm to take in account that if this ancestral node corresponds to the evolutionary correspond to a duplication, in this case, we always take the union and not the intersection. And this is a particular case because in EGT, we can economize evolutionary event by transforming a duplication by an EGT if there is a modification in genome labeling from ancestral node to children. So here we are. So here we follow the Fitch modified. So in this case, for example, you have duplication that could take union, duplication so take union. In this one, we see that there is intersection between 2 give 1 to 1, and we go down and go top, and after we have the possible ancestral genome labeling inferred by the Fitch modified algorithm. After, we have a top-down algorithm that so it is a second step also which Fitch modified algorithm. So here is like Fitch. The concept consists to go from the root and go to the children by minimizing the number of permutation. And if there is a permutation, you have the DLE distance increment from 1. So here at the root, if there is 2 labeling, you pick 1. Here we pick 0 to say we are in the mitochondria. After, we have to infer the evolutionary EGT event. So here, if we have a duplication and after we see that there is one of the children that is genome labeling that is different from the parent, so we have to infer an EGT. If there is a duplication, the duplication is transformed in an EGT copy that's allowed to economize 1 event. So here, if our lost event, we can, after, according to assignation, define if lost gene are localized on the nuclear or the mitochondria. We go down for the node where there is no modification, assignation between parent and children, there is no change. So here are 2 other cases where you have duplication that is transferred on EGT copy. And on the other side, so you have speciations that lead to two children with one children that is different genome labeling. So in this case, you have to create a new internal node that is where we insert an EGT cut, and this EGT cut you have to assign the genome labeling and to modify the genome labeling. And after this one, you have for the last one no difference aggregation. So we have resolved the DL-reconciliation. So in this case, it's based on the Fitch modified version of Fitch algorithm. So it only account for unitary cost. It doesn't allow to sample the different solutions that minimize the DL-reconciliation. So we developed a dynamic programming algorithm derived from Sankoff and Södergren's the dynamic programming algorithm, where we denote by dx bdx the minimum cost of a DL reconciliation of a gene tree TX. So on this algorithm, for leaf, if it's trivial if the inferred genome labeling is equivalent to the real genome labeling, so the cost is equal to 0, else it's infinite. And for internal node, we need to compute the minimum cost of a DLE reconciliation for 3 different evolutionary events, for the speciation, for the duplication, and for EGT copy. And we have recurrence formula for the 3. And according to which of the 3 recurrence formula minimizes the score, it will infer the evolutionary event associated to this node. So here is the formula for the recurrent formula of the dynamic programming, but I won't go into detail, but you can see in our paper. And at the end, so we have a dynamic programming that allow to sample all DLE reconciliations that minimize the number of duplication, loss, and EGT copy and cut. And also we can put arbitrary DLE cost on as a settings. After that, the designing of our algorithm, we designed a dataset to test the algorithm. So it was based on the dataset from Cannon et al. In 2014. They are from 2,486 complete mitochondrial genome. They make all versus all blast analysis to define a cluster of autologous group and that are based on mitochondrial protein coding genes. And after to have designed this autologous group, they after blast this group on the nuclear genome of the 2,486 species to find the mitochondrial genes that were located on the nuclear genome. So it corresponds to 34,000 protein coding gene sequence, and they were dispatched clustered in 140 cluster of autologous group that were called mitococs. So in our paper, we select a sample of the phylogeny. We take 11 plants because it was the group for which there was a bigger diversity between the repartition of mitochondrial gene on the mitochondrial genome and the nuclear genome. And we selected 28 mitocodes that correspond where there was in each of one mitochondrial genes that were present on the mitochondrial genome and on the nuclear genome. It corresponds to 326 protein coding genes, 184 encoded in the mitochondria, 142 encoded in the nucleus. There is the phylogeny. So to test our method, we test different parameter settings. So here as the notation for the cost, lambda for the loss, delta for duplication, rho is for EGT cut, when you have 0 if because you have a direction of an EGT endosymbiotic gene transfer, if it's from the mitochondria to the nucleus or nucleus to the mitochondria. And same for EGT copy with tau 0 and tau 1. So we define 5 cost setting. So the first cost setting is the unitary cost, or every cost has the same value. Settings 2 correspond to a basic setting where we have lower, higher cost for duplications than loss, a higher cost for endosymbiotic gene transfer compared to duplication, as it seems to is considered as rarer events than duplication. After that, we make some higher cost for the EGT copy compared to the EGT cut, because EGT copies there is a duplication then a transfer, while in EGT cut there is only a transfer of a gene. So this is the difference with setting 2 and 3. And after we also add higher cost for gene transfer from the nucleus to the mitochondria compared to mitochondria to nucleus, because there is almost no example of EGT transfer from the asserted, there is no asserted endosymbiotic gene transfer from the nucleus to the mitochondria. So we had higher cost for this too. And so we have the parameter setting S4 is S3 plus higher cost for the orientation, and the parameter S5 is parameter S2 with higher cost for orientation. So in summary, for the result, the same DLE-reconciliation with the 5 different settings. There is the same DLE with 5 different settings for 21 of the 28 mitocogs. For all the 7 mitocogs with more than one infer DLE-reconciliation, it leads to just 2 different DLE-reconciliation. And it's for our analysis, the parameter S4 seems to be the best parameter setting. Here is just 2 examples to illustrate that S4 parameter is the best setting. So here for mitocond 39, we observe the 2 topology inferred by parameter setting 1, 2, 3, 5, and the one by parameter setting 4. As we can see, if it's in blue, it means that the gene is localized on the mitochondria, and red branch represents it's located on the nucleus. So we observe that for the parameter 1, 2, 3, and 5, there is a reversion of the EGT transfer, that is a transfer from the mitochondria to the nucleus, and just after there is a transfer from the nucleus to the mitochondria. As I said, there is almost no validation of endosymbiotic gene transfer from the nucleus to the mitochondria. So this evolutionary scenario is very unlikely according to the literature, and S4 seems to be the best scenario. And for this second family, the mitocog 72, we have parameter setting 1, 2, 3 versus parameter setting 4 and 5. And in this case, we have an ancestral genome location that is in the nuclear for the first settings and in the mitochondrial for the 2 other settings. And we observe that for the first setting, you have 2 events of EGT transfer from the nucleus to the mitochondria, which is very, very unlikely. While in the second, they infer only one EGT transfer from the nucleus to the mitochondria, which is more likely. But so in this case, it seems that S4 is S5 are the best scenario. And for the other example I didn't show, S4 is always with scenarios that is the most on an agreement with scientist literature about EGT, endosymbiotic gene transfer. So in summary of our work, we provide the first algorithm to infer evolutionary history of EGT events. So it's an extension of the duplication and loss reconciliation. We add a genome location feature. We also add two new evolutionary events that are endosymbiotic gene transfer, cut, and copy. We provide a new dynamic algorithm to enumerate all possible DLE-reconciliations that minimize evolutionary event given an arbitrary DLE costs. The method has been implemented in the software Endorex program in Python and is available in GitHub repository at this address. From a perspective point of view, we would like to define an endosymbiotic gene transfer simulation model to provide EGT evolutionary history to assess the accuracy of Endorex DLE reconciliation inference. Also, there is an interesting question is about the elucidation of the root of eukaryotes according to EGT events. For now, eukaryote species tree is still unresolved. Here is a recent representation of the topology of eukaryotes tree. We see the main group for which it is well known that they are monophyletic, but we don't know exactly how they branch each other and what is the topology at the root of the eukaryote tree. So this with DNA sequence, there is a saturation of signal because it's very ancient event, and with the sequence it's not possible to resolve entirely the evolutionary route. So the idea is to use the DLE reconciliation to evaluate different eukaryote species tree topology. As EGT events are very rare events, it will be very useful to use the DLE reconciliation and find the topology for which we infer the minimum number of EGT events that could lead to a very good candidate for topology of eukaryote species tree. So I would like to thank my amazing co-authors, Nadia Almabrouk, Manuel Lafont, and Aida Wangrawa on the works done for gene tree and species tree reconciliation with endosymbiotic gene transfer. Also, a special thanks to Franz Lang that inspired us to on the algorithmic need to so a question of evolutionary history of eukaryote. I would like also to thank Mathieu Delab and Mathieu Gascon from DIRO for interesting discussion about endosymbiotic gene transfer during our meetings. And also I would like to thank all members of Cobios Lab that are very with a very interesting discussion and that helped me a lot during the development of this work. And finally, I would like also to thank the fundings from NSERC and FRQNT. Thank you for listening to my presentation.